Good day, this is Professor Will, CBMD PhD, case Thursday, June 13, 2019. It's 19:21:39 seconds Eastern Standard Time. This is a continuation of my scientific research, so let's hop to it here. Viruses found to use intricate treadmill to move cargo across bacterial cells. State-of-the-art technologies reveal bacterial cells organized like human cells, offspring insights for new phage therapies on untreatable infections. Date June 13, 2019. Source University of California, San Diego. Summary. Advanced technologies to explore the inner workings of bacteria. Biologists have provided the first example of cargo within bacteriophage cells transiting along treadmill-like structure. The discovery demonstrates that bacteria have more in common with sophisticated human cells than previously believed. Now using advanced technologies to explore the inner workings of bacteria in unprecedented detail, biologists at the University of California, San Diego have discovered that in fact bacteria have more in common with sophisticated human cells than previously known. Research laboratories have provided the first example of cargo within bacterial cells, transiting along treadmill-like structures in a process similar to that occurring in our own cells. It is not that bacteria are boring, but previously we did not have a very good ability to look at them in detail. With new technologies, we can start to understand the amazing inner life of bacteria and look at all of their very sophisticated organizational principles. Researchers analyzed giant Pseudomonas bacteriophage, also known as phage, a term used to describe viruses that infect bacterial cells. Research labs found that phage convert the cells they have infected into mammalian-type cells with a centrally located nucleus-like structure, formed by a protein shell surrounding the replicated phage DNA. In a new study, the researchers documented a previously unseen process that transports viral components called capsids to DNA at the central nucleus-like structure. They followed as capsids moved from an assembly site on the host membrane, trafficked upon a conveyor belt-like path made of filaments, and ultimately arrived at their final phage DNA destination. They ride along a treadmill in order to get to where the DNA is housed inside the protein shell, and that's critical for the life cycle of the phage. No one has seen the intracellular cargo traveling along a filament in bacterial cells before. The way this giant phage replicates inside bacteria is so fascinating. There are a lot more questions to explore about the mechanisms that it uses to take over the bacterial host cell. Opening the door to the new discovery was a research combination of time-lapse fluorescent microscopy, which offered a broad perspective of movement within the cell, similar to a Google Earth map perspective of roadways, in coordination with cryoelectronic tomography, which provided the ability to zoom into a street level. View that allowed the scientists to analyze components on a scale of individual vehicles and people within them. Researchers observed each technique perspective helped provide key answers, but also brought new questions about the transportation and distribution mechanisms within bacterial cells. Zooming in and out allowed us to observe a unique example where things just don't randomly diffuse inside bacterial cells. These phages have evolved a sophisticated and directed mechanism of transport using filaments to replicate inside their host that we could not have seen otherwise. Phage infect and attack many types of bacteria and are known to live naturally in soil, seawater, and human. Researchers believe the new findings are important for understanding more about the evolutionary development of phage, which have been the subject of recent attention. Viruses like phage have been studied for 100 years, but they are now receiving renewed interest because of the potential of using them for phage therapy. The type of phage studied in the new paper is the kind that one day could be used in new treatments to cure a variety of infections. If we understand how phages operate inside bacteria and what they do, the end goal would be that you might be able to start designing tailor-made phages for particular infections that are resistant. This is Professor Will, CBMD PhD, case Thursday, June 13, 2019. It's 19:51:13 seconds Eastern Standard Time. This is a continuation of my scientific research, so let's hop to it here. Salmonella-resistant antibiotics of last resort found in the U.S. Date June 13, 2019. Source, North Carolina State University. Summary. Researchers have found a gene that gives salmonella resistance to antibiotics of last resort in sample taken from a human patient in the U.S. The find is the first evidence that the gene MCR-3.1 has made its way into the U.S. from Asia. There are more than 2,500 known serotypes of salmonella in the U.S. Salmonella enrica 4-5-12I-ST34 is responsible for a significant percentage of human illnesses. The drug-resistant gene in question, known as MCR-3.1, gives salmonella resistance to colistin, the drug of last resort for treating infections caused by multi-drug resistant salmonella. In 2015, they saw that the MCR-3.1 had moved from a chromosome to a plasmid in China, which paves the way for the gene to be transmitted between organisms. For example, E. coli and salmonella are in the same family, so once the gene is on a plasmid, that plasmid could move between the bacteria and they could transmit this gene to each other. Once MCR-3.1 jumped to the plasmid, it spread to 30 different countries, although not, as far as we knew, to the U.S. Research labs generates whole genome sequences from salmonella samples every year as part of routine monitoring for the presence of antimicrobial resistant bacteria. Researchers did genome sequencing on 100 clinical human stool samples taken from the southeastern U.S. between 2014 and 2016. They discovered that one sample contained the resistant MCR-3.1 gene. The sample came from a person who had traveled to China 2 weeks prior to becoming ill with a salmonella infection. This project proved the importance of ongoing sequencing and surveillance. The original project did not involve this gene at all. The positive sample was from 2014, so this discovery definitely has implications for the spread of colistin-resistant salmonella in the U.S. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It's 20:39, 44 seconds Eastern Standard Time. And this is a continuation of my scientific research, so let's hop to it here. Fetal genome involved in triggering a premature birth. Study of more than 600 infant genomes points to pathway regulating neuron growth and inflammation. Date June 13, 2019. Source PLOS. Summary: mutations in the gene that codes for SLIT2, a protein expressed in fetal cells and placentas involved in directing the growth of the fetal nervous system, may contribute to the premature births, possibly by activating the mother's immune system. More than 10% of babies worldwide are born prematurely, which is the leading cause of newborn death, the source of lifelong complications. Preterm birth can run in families, suggesting that there are underlying genetic factors in the mother and the fetus that contribute to the problem. To identify the fetal genetic factors, researchers performed a genome-wide association study on a Finnish population that included 247 premature infants born before 36 weeks and 419 babies carried to term. The analysis pinpointed the variation in gene for a protein called SLIT2 that helps guide the growth of neurons during development and binds to a receptor protein called ROBO1. Using placentas, which are partly fetal tissue, the researchers showed that SLIT2 and ROBO1 were expressed at higher levels in placentas from premature babies compared to placentas from babies that were carried to term. Furthermore, in experiments using cell cultures of placenta tissue, the researchers found that ROBO1 plays a role in regulating several pregnancy-associated genes related to infection, inflammation, and immune response. The researchers proposed that SLIT2 and its receptor ROBO1 are part of the signaling network that causes spontaneous preterm birth, potentially by triggering inflammation and activating the maternal immune system. The findings dovetail with previous findings that the SLIT2 ROBO1 signaling pathways associated with multiple pregnancy complications, including preeclampsia and ectopic pregnancy. Researchers stated that our results are important as more detailed understanding about the fetal as well as maternal determinants triggering preterm birth will help us to identify those who are at the highest risk. This will allow targeted therapeutical interventions. In the future, researchers hope to validate these findings using a larger population. Such an analysis might also identify additional genetic factors that contribute to early birth. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It's 2100, 15 seconds Eastern Standard Time. And this is a continuation of my scientific readout. So let's hop to it here. Deadly tick-borne virus cured with experimental flu drug in mice. Bourbon virus has sickened people in Kansas, Missouri, and Oklahoma. Date June 13, 2019. Source: Washington University School of Medicine. Summary: an investigational flu drug cures mice infected with the rare but deadly bourbon virus, according to a new study. Bourbon virus was first identified in 2014 in a previously healthy middle-aged man in Kansas. The man arrived at the hospital with flu-like symptoms and a history of tick bites. Thinking he had ehrlichiosis, a tick-borne bacterial infection that causes similar symptoms and often affects people in the Midwest, doctors started him on antibiotics. But the man continued to decline, and further testing for ehrlichiosis and every other infection the doctors could think of came up negative. After his death 11 days later, a sample of his blood was sent to experts at the Centers for Disease Control and Prevention, CDC, who specialize in mysterious infections. The CDC researchers identified a new virus and named it bourbon, after the county where the patient had lived. Ticks have always been here, but now that we know that bourbon virus is in ticks in this area, there had probably always been people getting infected with bourbon virus, and we just did not know what it was before. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It is 21:11, 23 seconds Eastern Standard Time. And this concludes my scientific reading. I hope you enjoyed listening. Have a great day.